Good afternoon. Today is Saturday, June 13th, 2020. My name is Ricardo Sandoval, and this video is for week 5 of Biology 225, microbiology at Los Angeles Pacific University for the summer 1 term. This is lab number 8, and this lab is entitled Biochemical Testing Carbohydrate Fermentation. And here's just a quick little sign I made about this lab. So in this lab, what I'll be doing is I have 6 tubes of what's called phenol red mixed with nutrient broth. And phenol red, it's a water-soluble dye that's used as a pH indicator. It's red, given the name phenol red, and then given the pH of a given microbe, it'll turn either more towards the red or orangey side if something's acidic, or more towards the blue-purple side if something's basic. So what I'm going to do also is, beyond the phenol red, I'm going to use something called Durham tubes. They're these little test tubes I'll show you here in a minute. And these will actually go in the phenol red, and they are used to detect the production of gas by microbes. So since we're talking about fermentation, think about champagne, for example. Champagne is just wine that's been allowed to ferment, made from the Champagne or Champagne region of France. But after it ferments, it creates bubbles. So the same principle here. So if either the S. epidermidis or the S. cerevisiae cultures that I'll be using in the phenol red broths here do ferment, I will see bubbles in the Durham tubes. I will also be adding sugar powders to the Durham tube. So I have glucose, fructose, and mannitol to add. So glucose, as we know, is the sugar that's used in our bodies, what cells use to operate. Basically everything we eat becomes glucose, as it's a very good energy source. Fructose is the fruit produced by fruits. I'm giving the name fructose. Fructose is produced by fruits. It's what makes fruit taste sweet. And there's also mannitol. And mannitol is a sugar alcohol that's found in fruits and vegetables. Okay, so let's talk about supplies. So I have a total of 6 tubes of this phenol red that I'm going to use. So they're labeled number 1, 2, number 1, number 2, and number 3. So for each pair of tubes, so there's one for each sugar. So I have a pair of tubes called number 1 for S. cerevisiae and S. epidermidis. A pair of tubes number 2, S. cerevisiae, S. epidermidis for glucose. And then the last pair, number 3, is for mannitol. I may have the numbers wrong, but basically there's a pair for glucose, a pair for fructose, and a pair for mannitol. So, whoops, don't want to break them here. So, looking at number 1, this is S. cerevisiae for glucose. This is number 3. So number 3 goes for mannitol. So this is S. epidermidis for mannitol. This is S. cerevisiae for mannitol. This is S. epidermidis for glucose. This is S. epidermidis for fructose. And then lastly, S. cerevisiae for fructose. So number 1 is glucose, number 2 is fructose, number 3 is mannitol. I also have the powdered sugars in here. So this is mannitol. This is glucose powder and fructose powder. These are the Durham tubes that are going to go into the phenol red here. I also have a cup of alcohol with 2 pipettes that are sterilizing, and I'm going to put the 6 Durham tubes in the alcohol to sterilize right now. So, I don't know if you can see them very well, but they're in there, sterilizing. I have my S. cerevisiae culture, S. epidermidis culture, T-lights, and I believe that's it. So I'm going to finish putting these tubes away, put on my PPE, and then we will get started with the experiment. All right, so the first thing I'm going to do is I'm going to add the sugars to each of my pairs of tubes. So I'm going to start with adding the glucose. So again, I have my S. epidermidis glucose, S. cerevisiae glucose, and then I have my glucose powder. So what I'm going to do is I'm going to flame each of the tubes using my tea light. I will then pour half of the powder in each tube and then mix them. And then I'll do that also for the 2 tubes for the fructose and the 2 tubes for the mannitol. All right, so let's get started. So this is the S. cerevisiae one. Lid off. Just flaming it to sterilize. I'm gonna just put it in my test tube rock here just for ease of pouring. So this is, again, this is my glucose powder. I'm gonna pop the lid off and pour half in. That's about half. A little bit more than half, but it is what it is. So I'm gonna flame this again to sterilize. Put the lid on. And I'm going to shake it, well, invert it a couple of times, just to make sure that the glucose powder really dissolves. Okay, we're gonna set this aside for now. This is the S. epidermidis tube. Lid off. Flame the lid, the tip of the tube to sterilize. Back in my test tube rock. And the remainder of the powder is going to go in the tube. Okay. This is now going in the trash. Gonna flame this again. Okay. That's been flamed. Lid on. Invert a couple of times to sterilize, to mix. Okay, so that's the glucose. Next up is fructose. Again, a sugar produced by fruit, that's why fruit is sweet. So, fructose. And this is my S. cerevisiae fructose tube. Lid off. Again, I'm going to run the tip through the flame to sterilize. Okay, that part's done. And again, half of this is going to go into the tube. And quite interestingly here, the crystalline composition of the fructose looks somewhat similar to sugar that we use, granulated sugar, to bake and add to coffee, for example. A little bit more. And since the crystals are a bit more coarse in comparison to the glucose, it is easier to pour just in half. Okay, in just half of the solution. So, of the granules, I mean. So that's that part. That's flamed. Lid. And let's invert a couple of times to mix properly. Again, because the granules here are much larger than the glucose, so I just want to make sure they really do dissolve well and not stick to the side of the tube. Okay, so that part's done. We're going to do the same thing for the S. epidermidis fructose tube. Lid off. Running through the flame to sterilize. Back in the test tube rock. The remainder of the fructose, or the other half, goes in here. so that goes in the trash. Sterilize again by running it through the flame. Okay. Lid on, and then invert several times to mix. Okay. And then lastly is the mannitol. So my last two tubes. Here's the mannitol, which again is powder very similar to what the glucose looks like. This is the Cerevisiae one. Running it through the flame to sterilize. Okay, back to the test tube rock. Lid off the mannitol. And then again, half goes in here. Okay. Flame the lid to sterilize. Lid on. Invert to mix. Okay. And this is my epidermis mannitol. Flaming to sterilize. Remainder of the powder goes in here. Okay. Garbage. Flame again. And then invert to mix. Okay. All right, so the next step now is to put a Durham tube, that little test tube looking thing that traps gas if gas is produced by the fermentation reaction. So I've removed all 6 tubes from the alcohol and made sure that they're nice and dry. Make sure all the alcohol is drained. So I'm just going to put one in each tube. They're going to go face down. So the opening of the tube is going to go down into the tube with the rounded bit facing up. Okay, so that looks about right. I don't see any air trapped into the tube. So there's one. Now at this point, the numbers of the tubes don't matter since I'm not adding anything into the tubes, just the tubes themselves. 2. This one has air bubbles, so let's move the tube around until the air bubble's gone. Okay. 4. hopefully you can see this on camera. So there's 1, 2, 3, 4, 5, and 6. Okay, the next step here is I'm going to add the S. epidermidis and the S. cerevisiae cultures into the appropriate tubes. So what I have here in the front, this is my S. epidermidis culture, and then my 3 phenol red tubes with the glucose, fructose, and mannitol. So I'm going to take one of my pipettes here, squeeze out all of the alcohol, and just bring it to get any residual alcohol off. Okay, so that should be good. So I'm going to flame my culture lid, to sterilize it using my tea lights. Okay, so that part's done. And then 4 drops of the culture are going to go into each tube. So here's the first one. I'm going to flame it. Okay. With my pipette, I'm going to draw up some of the culture. And this is the one, this is S. epidermidis fructose, 4 drops. 1, 2, 3, and 4. Okay. I'm going to flame this again. Lid on. And that's that. Okay. Same thing for the next one. Lid off. Flame to sterilize. And this is glucose, 4 drops. 1, 2, 3, 4. Okay. Flame again. Okay. Lid on, and that one's done. And then the same thing for the last one. This is S. epidermidis mannitol. Lid off. Okay. 4 drops. 1, 2, 3, and 4. The remaining culture is going to go back into the tube. Okay, this is going into bleach. Flame this again. Okay. Lid on. I'm going to flame my culture tube here to sterilize. Okay. Lid on, set it aside. And I'm going to do the same thing for the S. cerevisiae cultures. The S. cerevisiae phenol red tubes with the different sugars. Okay. So this is my S. cerevisiae culture. I'm going to remove this other pipette from the alcohol, squeeze out the alcohol completely. And just make sure that it's incredibly free of alcohol so that it doesn't kill the bacteria, the microbes. Okay, there's that. So again, this is my Cerevisiae culture. I'm gonna take the lid off. Flame the lip of the tube to sterilize. Okay. Alright, and I'm going to start with the Cerevisiae mannitol tube. Lid off. Flame to sterilize. Okay, I'm going to drop some of the culture with my pipettes. And again, 4 drops. 1, 2, 3, 4. Alright. Flame this again. Okay. Lid on. And set it aside. Next one is a Cerevisiae with fructose. Lid off. Flame to sterilize. Okay, and again, 4 drops. 1, 2, 3, and 4. Flame to sterilize. Alright, lid on and set aside. And then lastly, this is my Cerevisiae glucose tube. Lid off. Flame to sterilize. Okay, 4 drops. 1, 2, 3, 4. The remaining culture is going to go back in the tube. This goes into my bleach bucket. I'm going to flame this one last time. Okay, lid on. I don't know if I mentioned at the inception of this video, but I did allow everything, the phenol red, all my cultures out of the fridge for a good 45 minutes to an hour just to come to room temperature. I'm going to flame my culture tube. Okay, so this is the conclusion of the experiment video for Lab 8. These phenol red cultures are now going to sit in my incubation location, which is the dark temperature controlled closet of my home office for 12 hours. And then we'll reconvene from there.